Okay. So, video two. We're going to try and go pretty quick through this one because the last one was a long one. This one, we're just going to look at the different distances that cell signal acts on. We'll be talking about hormones a bit as well. So, first thing, we want to talk about the evolution of cell signaling, like why would our cells talk to each other in the first place? But single cell life still does communicate with each other. Yeast that you can see there uses a ligand receptor system to do mating types. So that's how they tell if, essentially, it's not male female, but same idea. So yeast communicates so that it can mate. Other bacteria will communicate so they know, like, it's a little crowded over here and it's less crowded in some other area, so it helps them not overpopulate in certain regions. But ultimately, this was useful from the very beginning. Just like it's helpful for humans to talk to each other, it is helpful for single cells to talk to each other as well. But we are going to talk much more about our fancy animal human signal pathways. Main idea to keep in mind through this is that cells can only respond to a signal if they have the specific receptor for the signal. So you're not going to see insulin receptors in your brain, because why? Insulin shouldn't be there. Your brain's job is not to store glucose. So it will not have insulin receptors. So few different types. We're going to talk a bit about local signaling and then more long distance signaling. But the types being direct contact, the cells are actually attached to each other that you can see up there, with their membranes or cell walls actually sharing portions of them. It can be cell-cell recognition, where it's just a surface protein and a receptor protein binding to each other, so still attached but not sharing a membrane. You can have hormones, which is your long-distance signaling. Those will travel through your bloodstream after being secreted. And then you'll have local regulators, which is short-distance signaling. We're still secreting the ligands, but they're not actually going to the bloodstream. So let's talk first about direct contact. You'll often see this called juxtacrine signaling. If you know the word juxtaposition, that's like a contrast that you get by having two things next to each other. So juxta means like juxtaposed is like being next to each other. And this is just signaling where you need direct contact. There is no secretion of ligands. They can just exchange material between the two. So in animals, this is gap junctions. In plants, it's plasmodesmata. And we're just directly sending material back and forth. It can also be through membrane reception, again, through those surface proteins binding to surface receptors, but not actually sharing any membrane. Short distance, using local regulators. This can be either autocrine or paracrine. Autocrine would be a bit less common. Essentially, autocrine is self-stimulating, self-signaling. You release your ligand and it goes out into the environment and binds to your own extracellular receptor to cause a response. You might be thinking, why the heck would this ever happen? What's the point? Just do the response inside the cell. Cancer cells will abuse this. They can secrete growth factors that will then bind to their own receptors and then cause the cancer cell to keep dividing and growing quickly, so kind of hijacking it. But paracrine signaling is just much nicer, just chemical signals you secrete, pass them off to a nearby cell. This is exactly what our synapses do. They're just designed to do the paracrine signal efficiently. And then I have that slide twice for some reason. So let's talk about a short distance example that's pretty cool. So our immune systems have what's called an adaptive immunity, meaning we can target specific antigens of all different types, and we can make and tweak our immune system's response to those specific antigens. And antigens are just anything that's going to cause an immune response, so like bacteria, virus, all that. So what happens is, initially your immune response gets started up by a special white blood cell called a helper T cell, which is going to have a bunch of membrane receptors on it to just bind to different antigens. The idea being just have a bunch of different shapes and hope that you can bind something. They become active if they actually do bind to an antigen on an antigen-presenting cell. So an antigen-presenting cell is a macrophage. A macrophage is a big cell that eats other cells. It's part of our immune system. And they just eat the antigens, maybe eat the actual bacteria, chop them up, and put some of their fragments on their cell surface so that they can be identified by the wandering helper T cells. So the next step is our helper T cell is going to bind to the APC. This will be a type of juxtacrine signaling. And they're just going to release a bunch of signal molecules called cytokines. Both cells will release cytokines. This is an example of paracrine signaling. And then the T cell itself is going to release its own set of cytokines with a different signal that tells itself to proliferate. So this is autocrine signaling. The purpose of this is this T cell has the correct antigen receptor. So this T cell knows what the bad guys look like. So the antigen presenting cell is first saying, yes, I have a match. You're a match. Great. And then that's when it releases its cytokines. And then the helper T cell says, great, I have a match. I'm going to tell myself to multiply so that we can find all of these antigens all over the place. So then the T cells, now that there's a bunch of them, are going to go and activate other white blood cells that are going to now have more targeted attacks. They are just going to bind to the antigens and kill them without a need for the helper T cells involved. So helper T cells are kind of like the guards patrolling around. They recognize a bad guy. They call in the troops. The troops do the job. Long distance communication is going to be referred to often as endocrine signaling. Your endocrine system is basically your hormone system. And it's long distance because it involves secreting hormones into the bloodstream. So this basically lets cells of one cell type talk to cells of another cell type. It's how, for example, your brain can talk to cells in your pancreas or your liver without using your nervous system. So this is for slower, more like homeostasis-like changes. Hormones are just chemical signals in your blood. So it's a pretty general term. Most hormones are proteins, but the two hormones you probably know the most, testosterone and estrogen, are steroids. Lipids, they can enter the cells on their own without need for a receptor. So we have a bunch of different glands that are going to secrete all of these hormones into the blood. They're spread all throughout the body. And then the hormones will travel through the bloodstream to target cells in other parts of the body. You can imagine that with your sex hormones being small and nonpolar and being able to enter any cell, they're going to be in pretty much every cell in your body that has access to the bloodstream. And so they can cause a huge range of effects because they don't need a receptor on the surface or anything. They can just get into the cell on their own and start causing havoc. Another one that's long distance that we know, insulin. It's a hormone secreted by the pancreas, and its target cells are fat, muscle, and liver cells. It tells them to essentially, there's too much glucose in our bloodstream, absorb some of this and store it, either as fat or glycogen or something like that. So insulin just binds to the receptors on those cells and tells them to bring the glucose inside using some glucose transport proteins. Then depending on what type of cell it is, they'll have different uses. So fat cells will turn it into fatty acids and store it as fat. Liver and muscle cells will make glycogen, which is more for, it's like medium-term storage. Glucoses are super short-term energy source. Glycogen is like medium, and fat is long-term. So again, one stimulating molecule, one ligand, multiple different effects depending on the tissue and depending on the receptors. The last one I'll talk briefly about is human growth hormone. This will be abused often by athletes to gain a competitive advantage. It just stimulates bone and muscle growth by increasing their cell division. So it just makes your muscles grow bigger. And it essentially just tells your fat cells to break down fat and release glucose, and use that glucose to divide. Dividing takes a lot of energy. And so you're just kind of artificially telling your body to grow that muscle without a stimulus for it. The pathway is super complicated. I'm not going to go into it too much. It's multiple effects in all sorts of different cell types. Sometimes you just have effects that are inhibiting other effects. So it's very interesting. We don't have the time to go into it, but it is very, very complex. This is a simpler form of it that you can see. It's essentially a phosphorylation cascade with an RTK complex that just tells us to make proteins so we can grow and divide. Okay, that is the end of this video. Our next one will be on homeostasis and feedback. Hopefully this one wasn't too bad. I'm going to try and keep them shorter than the first one, but that's all. See you later, skaters.